Hello, everyone. What we're going to be talking about in this lecture is the last group of biomolecules, which are referred to as nucleic acids. So what we'll discuss here in this lecture is, first of all, we'll define what the building blocks, nucleotides, and nucleic acids are and what is their function in living organisms. And then we'll get first into the building blocks, so the nucleotides. We'll talk about their structures and the different types you can find, which then lead to the formation of the two types of nucleic acids, which we'll discuss later on in the lecture. So we'll talk about their structure, their definitions, the types, as I say, either DNA or RNA, basically, and then we'll get into the differences in structure and common parts of the structure as well. So nucleic acid and nucleotide, I stress this slide refers to both types, nucleotides and nucleic acid altogether. So nucleic acids are polymers of nucleotides. Okay, please keep them different. Some of you confuse or use the terms nucleotides and nucleic acid as the same thing. No, nucleic acids are made by several nucleotides joined together. You'll see later on. Now, when it comes to function, so nucleotides and nucleic acids have different functions. We perfectly know, we talk so much about it, how nucleic acids are important for the storing of genetic information. If you remember, in the RNA world, everything started with RNA, and then the storing of genetic information was later moved to a second type of nucleic acid, which is DNA. Then we know how they're important for translation of information from DNA to proteins through RNA, through a process of transcription first and then translation. Nucleotides are important for energy storage as well. You know, ATP particularly, you see when we talk about different metabolic processes such as cellular respiration, photosynthesis, and also when it comes to movement of material across the cell membrane, how ATP particularly, GTP as well, but ATP particularly, so one of these nucleotides that make up actually RNA, are important to store the energy in the phosphate bonds there, to store it and have it quick energy to be used. They can work also as coenzymes. You see when we talk about cellular respiration and photosynthesis, how some modified nucleotides, they actually help enzymes to temporarily store the energy in the form of electrons. And then finally, some nucleotides, modified nucleotides, such as cyclic AMP, can work as messenger inside the cell as well. If you look inside a cell, you can actually see that the concentration of cyclic AMP is in every fraction of a second, changes all the time, and the change in concentration have different effects inside the cell. So the cyclic AMP work as a messenger within the cell, or even among neighbor cells that are joined together through different type of ionophores. So let's start with the nucleotides, talking about structure. First of all, there are two different types of nucleotides, which gives origin to the two different types of nucleic acids. So when it comes to the nucleotides that make up RNA, so ribonucleic acid, we talk about ribonucleotides. There is a structural difference between them. Whereas when we talk about nucleotides that make up the ribonucleic acids, or DNA, we talk about the oxyribonucleotides. Despite their difference, they all share common features when it comes to the generic structures. What are these common features? Well, they're all made by the same three components, generic components at least. They all contain something that we call the nitrogenous base. Here in green, you can see it. This is referred to oftentimes as nitrogen-containing base 2, or nucleobase as well. This is attached to a pentose, which is the second component of all nucleotides. When we talk about the component of nucleotides made up by the pentose and the nitrogenous base, you refer to this as the nucleoside. So the difference is in this letter here, nucleoside versus nucleotide. And a nucleotide is basically a nucleoside which has the third component attached to this, which is one or more phosphate group attached to that, 1 to 3 phosphate group. So Whether you're talking about ribonucleotide or deoxyribonucleotide, they will all have these three components in them with some differences in there. Now, starting from the pentose. So the pentose here is that component or nucleotide that joins together the nitrogenous base to the phosphate group. It's in between there. Now, these pentose, so it's a 5-carbon monosaccharides, can be of two types. If you're talking about the nucleotides that make up RNA, so ribonucleotides, it will be ribose. If you're talking about those nucleotides that make up DNA, so deoxyribonucleotides, it is deoxyribose. The difference between them is in what is attached to the carbon 2 of these pentose. So in ribose, you would have an hydroxyl group attached. In deoxyribo, instead, you have only a hydrogen. So there is one oxygen basically missing. Here you have only a hydrogen, which is not specified in this type of structure, but it's there. So the difference is in an oxygen, hence the name deoxyribonucleotide or deoxyribose for the pentose itself. So deoxyribonucleotides would have these pentose in it. So deoxyribose, ribonucleotides that have ribose. Notice that both of them are in the beta form. Other important thing to remember, other than the difference in what is attached on carbon 2, is that the nitrogenous base, no matter what pentose you're talking about, will always be attached to the carbon 1 of the pentose, whereas the phosphate group is always attached to the carbon 5. And the bond between different nucleotides is always mediated by the carbon 3 and the phosphate group in carbon 5. We'll see in more detail in the next few slides. Now, when it comes to pentose, the numbering is always specified with a prime symbol. So these will be the carbon 1 prime, carbon 2 prime, carbon 3 prime, 4, and 5. It's done clockwise in this picture right there. Now, the reason why I use the prime symbol is to distinguish these elements from the elements that are present in the nitrogenous base. For example, if I'm talking about carbon 2 prime, you're talking, and I'm talking about this carbon in the pentose and not in the carbon 2 in the nitrogenous base. So prime just allows to distinguish between them. Now, together with this sugar, so the pentose, which again can be different in ribonucleotides versus deoxyribonucleotides, on 5 prime of the pentose, no matter what type you're talking about, you always find 1, 2, or 3 phosphate attached to them. Now, if you have only one phosphate group, you're talking about a nucleotide monophosphate. If you have 2 of them, you would talk about like it. So here, this will be adenosine monophosphate, AMP. This has 2 of them. We refer to a nucleotide diphosphate, so adenosine diphosphate. And if you have 3, you're talking about adenosine triphosphate in this case, which is that molecule which is used to store quick energy. Notice one thing, I'll repeat the same thing over and over, but it's a good way to fix the concept, is how ATP as an hydroxy group on the carbon 2 prime here of the pentose. So this is ribose, not deoxyribose. Hence, this is a ribonucleotide. It's one of the 4 nucleotides that makes up RNA. And this is the one that is used to quick release energy by separating one of these phosphate group here. So converting ATP into ADP, the energy of this bond is somewhat released. It's not really released, but it's transferred. You see, when we talk about different processes, we talk about movement of material across the membrane or phosphorylation of substrate and so on. The energy is transferred from this molecule. So the energy that was stored here is transferred to some other molecules or process by converting ATP into ADP. Third component of all nucleotide is the nitrogenous base, which is always attached to the carbon 1 of the pentose, whatever it is. And it can be of 2 main groups, different types. So there are 5 possible nitrogenous base. You have cytosine, thymine, adenine, guanine, or uracil here. So cytosine, thymine, adenine, and guanine are found in DNA, and cytosine, adenine, guanine, and uracil are found on RNA. So basically, cytosine, adenine, and guanine are commonly found in both ribonucleotide and deoxyribonucleotide, hence in RNA and in DNA, whereas thymine is found only in DNA and uracil only in RNA. So thymine is found only in deoxyribonucleotides, uracil only in ribonucleotides. Now, based on whether they're made by a single ring or two rings, these nitrogenous base can be divided as pyrimidine if they have one ring, or purine if they have two rings. Now, moving on to the higher level here, let's start talking about nucleic acid. So nucleotides, which are made by these three separate components, are the building blocks of nucleic acid. So nucleic acid are basically polymers of nucleotides joined together through, you see later on, what is called phosphodiester bonds. Now, there are two types of nucleic acids, which are formed by two different types of nucleotides. So if you're talking about ribonucleotides, so these nucleotides that have ribose in them, now you're talking about the polymer of ribonucleotides is referred to as ribonucleic acid, or RNA. Make sure that you remember the full name. Then if you're talking about a polymer of deoxyribonucleic, there's a mistake, a polymer of deoxyribonucleotides should be here. You're talking about deoxyribonucleic acid, or DNA. So made by those nucleotides that have deoxyribose in them. Now, this polymer is referred to as a strand. When you talk about a strand of DNA or strand of RNA, you're talking about a polymer of nucleotides which are joined by a specific type of bond, which is referred to as phosphodiester bond. The naming here comes from the fact that this bond, which is mediated between the carbon 3 of one nucleotide and the carbon 5 of the following nucleotide, is mediated by a phosphate group. So it has two ester bonds. There will be an oxygen here and an oxygen here. This schematic doesn't show it. But there's an oxygen here, an ester bond here, an ester bond here. It's two ester bonds mediated by a phosphate group, hence the name phosphodiester bond. Okay. Now, these polymer of nucleotides joined by phosphodiester bond leads to the formation of what is called the backbone of the nucleic acid, which consists of the alternation of the phosphate group attached to the pentose. In this case, this will be ribose. Okay, this will be an RNA strand. Phosphate, pentose, phosphate, pentose, phosphate, pentose. This structure here is referred to as the backbone of the nucleic acid. And then attached to the backbone, you have the nitrogenous base that do stick out from the backbone itself. Another important thing to understand here is that every strand has a specific orientation, which is referred to as 5' to 3' or 3' to 5'. When you talk about orientation, you talk about 5' referring to where the carbon 5, the last carbon 5, is found. So where the last carbon here in this strand, so if you look at this strand, the last carbon you find here is a carbon 5, will be a carbon 5'. So this is referred to as 5' of the strand. Whereas at the end of this strand here, the last carbon you find in the backbone is carbon 3'. Hence, this is called the 3' end of this strand. And then the direction here is 5' to 3', or 3' to 5'. Why is this important? You see, when we talk, for example, about DNA replication, you see that DNA polymerase, for example, DNA polymerase 3, can make a new strand, can build a new strand only in the 5' to 3' direction. What does it mean? It means that it will get this strand to grow. Let me just use, in this case, I'm going to use the pen for a second. It will get it to grow in this direction. If it works 5' to 3'. So this being 5' and this 3'. So what it means is that basically it can add the next nucleotide only to this hydroxyl group. So it would add the next one here, and then the next one to the hydroxyl group of the nucleotide that I just added. So getting it to grow in this direction. So it cannot add any nucleotide to the phosphate group. Otherwise, it will be growing the strand in this direction, which will be a 3' to 5' direction. Okay. So that's the reason why it's important to understand. It's important also to understand when we talk about anti-parallel strands forming the alpha helix. We'll get there in a second. Now, these strands, whether you're talking about DNA or RNA, this happens in both of them. These strands can actually be joined together. So two strands, two of this, can actually be joined together through the formation of hydrogen bonds, not within the backbone, but within the nitrogenous base that stick out from the backbone. So these bases here can form hydrogen bonds with other nitrogenous base from a different strand. And this takes place through a purine to pyrimidine base pairing, which is very specific. So what you have is that adenine, so this type of nitrogenous base, can form hydrogen bonds only with thymine when it comes to DNA, or uracil when it comes to RNA. And it can form only 2 hydrogen bonds. Whereas guanine can form hydrogen bonds only to cytosine, not to adenine, thymine, or uracil. So the pattern here is between adenine and thymine, or vice versa, or adenine and uracil, or uracil and adenine, and guanine to cytosine, cytosine to guanine. So in this case, again, guanine and cytosine can form 3 hydrogen bonds, and adenine, thymine can form only 2 hydrogen bonds. And that's the reason why double-stranded DNA molecules that have a lot of adenine, thymine in them, they're easier to open up compared to sequences that have guanine and cytosine, because in the end, you end up having more hydrogen bonds between 2 strands, which are, so if you have a lot of adenine and thymine, you have less hydrogen bonds compared if you have a lot of guanine and cytosine in them. And you see, this is very important when it comes to DNA replication that starts at the origin of replication. So sequences that are rich in adenine and thymine, not surprisingly, so easy to open up. Or even in some promoters, those sequences that promote the transcription of DNA into RNA in the genes are rich in adenine and thymine, easier to open up. You need access to the strand to be copied. Again, we'll talk about that in more detail when it comes to DNA replication and transcription. But keep that in mind, sequences that are rich in adenine and thymine are easier to break apart, to separate. So 2 strands attached by adenine and thymine are easier to separate compared to those rich in guanine and cytosine, because they have overall less hydrogen bonds in between them. Now, as I said before, now the RNA structure can take place, or this formation of these hydrogen bond can take place both in DNA or RNA. The difference is that in both cases, this takes place through an anti-parallel type of bond between the 2 strands. What we mean is that the 2 strands that form the hydrogen bonds would have opposite direction in terms of 5' to 3' compared to each other. Look at the DNA here. DNA is formed by 2 different strands, as I said before. So these are 2 separate molecules. If you follow here, this is the 5' of this first strand. Will go like this. You can follow the highlighter here, and it ends right here with a 3'. And this is a separate strand here, right? Now, the first one is in 5' to 3' orientation, the other one in 3' to 5'. So what it means, practically speaking, that here you will find a phosphate group attached to the 5' carbon of the oxyribose here, and here you would end up with an hydroxyl group. The opposite here, you have an hydroxyl group found here and a phosphate group here. These 2 strands are oriented anti-parallel to each other, hence they form these hydrogen bonds, which then lead to the alpha helix that you see there. This happens also in RNA. The difference is that you don't have 2 separate strands. If you look in RNA, you have partial double helixes formed by the same strand. Follow this. If you see, this is the backbone in blue. You see that it's a single backbone here. So it's actually nitrogenous bases in 2 different parts of the same strand that do form these opposite anti-parallel bonds. So in this case too, if you look, starting from here to here on this part of the strand, it will be 5' to 3'. And then these coils, and at this point you are 3' to 5' in this orientation here. So anti-parallel to the other one. It's more similar if you think to what happens in proteins. If you remember, the formation of the beta pleated sheet, for example, could take place the same way. Same strand, like folding and forming hydrogen bonds. Same thing here when it comes to RNAs. The same strand leading to the formation of hydrogen bonds between adenine and uracil in this case, and cytosine and guanine, leading to the formation of a partial double helix. Another important thing to remember is that, like in alpha helix, in proteins, these coiling is what is called right-handed. Sometimes you find pictures on DNA put as a left-hand helix. It's not true. It's incorrect. DNA exists, or even the partial double helixes in RNA, they're always right-handed. So, which shows you how it's supposed to turn based on following, basically. If you put your hand and look how it's supposed to go upward, it will be following the shape of your right hand, not your left hand. This is an example of how partial double helixes are formed in RNA. Oftentimes, I get students to comment that, oh, double helix is formed only in DNA. No, it's not true. They can be formed in RNA molecules as well. The difference is that there are only partial double helixes, so it's not the whole molecule that it has double helix. And as I say before, it's formed by the same strand. So it's the same strand. Here you see the structure, a generic structure of a tRNA, one of these RNA molecules that takes the amino acid to the ribosomes. We'll talk about them when we talk about translation. The amino acid will be attached here. There's an anticodon here, but beside that, these molecules, you can see, the same strand here can form some partial double helixes here. They have complementary sequences here, which then lead to the formation of partial double helixes that look more like this. You can see in real 3D shape. Okay, so this will be where the 3' hydroxy group is in here, and this will be where the anticodon is actually present. Now, when it comes to double helixes, as we say, they're all right-handed here, and they have specific properties. Whether you talk about DNA or RNA, they're very similar, the properties. This structure here of the double helix is the one that was proposed by Watson and Crick in 1953. I'm sure you're aware of that. And I want to stress, I use the word propose, not discovered, because in reality, what Watson and Crick did, they just brought together discoveries from other scientists and they put them together, coming out with this structure, with this model. But there are many other scientists like Chargaffs, Franklin, and so on that contributed overall to this model. Just Watson and Crick put together their discoveries. And so, as we say before, double helix are always formed by two anti-parallel strands. They're formed by the hydrogen bonding between the nitrogenous bases, and these lead to the formation of an alpha helix right-handed, which has what is called a major groove. When you talk about the groove, you're talking about the spacing between the backbones. So the backbones here, the alternation of phosphate, deoxyribose in this case, phosphate deoxyribose here, the spacing here between the backbone can be larger in some parts, so that's referred to as a major groove, and smaller in other parts, so which alternate with each other. This is referred to the word smaller to the minor groove. Again, the bonding is mediated by base pairs, so with the nitrogenous bases here in between, in the middle here, and which is done by the nitrogenous base to maximize the strength of these hydrogen bonds. They're actually parallel to each other. They form a plane. If you look here in better detail, you can see how these bases are basically all forming almost like some sort of ladder, which maximizes the strength of the hydrogen bonds between them. Okay, and final detail here is per turn you have about 10 to 11 base pairs. So every turn, so going, if you go from here to here, the same point in the helix, you could count 10.5 bases in average. Okay. So summarizing here, this lecture is basically done. What we talked about here is nucleotides and nucleic acids. So first we define what nucleic acids are and the function of nucleic acid and nucleotides. Then we started with structure nucleotides. We talked about the generic structure shared both by ribonucleotides and deoxyribonucleotides, and then we discussed the differences between them. So the difference in the pentose sugars with ribose present in ribonucleotides and deoxyribose present in deoxyribonucleotides. And then we talk about the difference in nitrogenous base found in ribonucleotides, where you have adenine, guanine, and cytosine sharing both of them, but ribonucleotides would have uracils, whereas thymine will be present only in deoxyribonucleotides. And then we move to nucleic acid. We talked about their structure, so how the strands is formed basically by polymerization of nucleotides joined together by phosphodiester bonds. This leads to the formation, and discuss what a backbone is, the sequence of phosphate, pentose, phosphate, pentose, and define also a specific orientation, which is referred to as 5' to 3', with the naming coming from the last carbon in the pentose that you can find at one end or the other, with the 5' ultimately having a phosphate group attached to the carbon 5' of the pentose, and the 3' having an hydroxy group attached to the carbon 3' of the pentose. Then we discuss also how the double helix can be formed both in DNA or partially in RNA as well, through the purine-pyrimidine hydrogen bonds with specific pattern. So where you have adenine binding to thymine in DNA or uracil in RNA, and cytosine binding only to guanine, both in DNA and RNA. And finally, we discuss, we showed the structure of the double helix both in ribonucleic acid and deoxyribonucleic acid, so DNA, RNA, sorry, I should say, and DNA respectively.